Hello, I'm going to be talking about the toolbox we just released alongside our OHBM poster, which is entitled VB Toolbox, a tool for investigating neural feature gradients on the brain in Python and MATLAB. Now this toolbox allows us to create gradient maps which let us look at large-scale interareal differences and their relationships in the brain, typically on the cortex. And it also allows us to investigate and quantify boundaries between brain areas, and this is the VB index, which is an index we've recently proposed in a preprint, which you can find linked to our poster. So I will be mainly focusing on the Python version of this program because it is the easiest to install. No Python is really needed to understand how it runs, and it's a simple command line toolbox program that anyone can use and install. Okay, so the first thing I'm going to do is I'm going to create a virtual environment, and I'm going to do that with conda create, and that's just creating a virtual environment. And we can just find the activate, and now we've moved from our base environment to our VB tool OHBM. And this, for people not used to using virtual environments, is so that we can install a separate installation of VB Toolbox and not interfere with any other program. Okay, so now we just need to install the program using pip. So pip install VB Toolbox, and that should run fairly easily. And it will install all of the dependencies you need. Okay, and that's VB Toolbox version 1.1.0 installed. Okay, and let's first VB tool. If we do VB tool and help, that will give us our help file, and it should explain everything over here, how to use the program. It will send you to the GitHub if you're interested in looking at the code, exactly what the toolbox does. Incidentally, the GitHub also houses the MATLAB version. Instead of Py VB Toolbox, it's MATLAB VB Toolbox within the same VB index account. And you can go there for either of those two and look at the code and experiment for yourself. It is licensed under a GPL license, so you can feel free to use it under the terms of the GPL license. And these are the important bits. So the command is VB tool. That's how you run the program. And then we require 3 arguments. The first one is S, which is the surface mesh. This can be any surface mesh of the data set that's associated with the data set you're interested in, and this is a GIFTY file. The data file needs to be another GIFTY file that is associated with that surface. And it's important, unfortunately, at this point, all data has to be in GIFTY format. SIFTY is not accepted as a format. And then finally, we need the base name for the output. And then we have a bunch of other optional commands, some of which are needed for certain analysis. And there are 3 main types of analysis that can be run. So the first type of analysis is the so-called searchlight VB index. And that runs a searchlight. It outputs a map which you can sort of think of as an edge detector. And more details in the preprint. I will not go into exactly what the code does and what the meanings of the maps are, but if you go to the preprint and you find the searchlight VB index, well, that's one of the analyses that you can do with this software package. The second one is the whole brain gradient. So this is the typical, now gradient analysis is becoming more and more popular, and this is more or less the typical gradient analysis that is run. Again, I'll refer you to preprint for certain minor differences between different software packages and implementation of the gradients. And the third type of analysis is, well, a sort of hybrid where you can do gradient analysis of clusters. So let's say you already have a parcellated image of the brain. Let's say in this case we're going to use the HCP multimodal parcellation. You have a bunch of predefined parcels that you're interested in, and you compute the gradient in each of those parcels and the associated VB index for that entire parcel. Again, if you want to know what the VB index for that entire parcel means, please have a look at the preprint for more details. Okay, so with that little background, we can explore the optional arguments. So we'll start from the bottom because that is actually the more useful one. So this is the C flag is only necessary if you are interested in doing the clustered version. So let's say in this case it would be your MPM, your multimodal parcellation. Right? Again, this needs to be a GIFTY file. It can't be a label file that HCP gives you. You need to convert that into a GIFTY file where each vertex contains an index. So let's say parcel 1, all of the vertices need to be labeled 1 and 2 and 3 and 4, whatnot. So that's the cluster file. Clusters with index 0, however, will denote the medial brain structures, so the corpus callosum, the middle of the brain that is usually removed from an analysis. So anything in your cluster file that is 0 will be effectively removed from your data. Okay, the second file, which is closely related to the C flag, so the M flag, this is done when you're doing, this is useful when you're either doing the full brain gradient or when you're doing the searchlight VB index, and this defines a mask of where your cortical vertices are. Effectively, again, this is very similar to the clusters, but consider the whole brain as one cluster, and the 0 values again denote the midline structures. The FB flag is needed if you want to create a full brain gradient. This is by far the longest analysis. It takes quite a while to run this because it's the analysis that's hardest to parallelize. The norm with the N flag allows you to set various normalizations of the Laplacian. You by and large can not fiddle with this, and you can be happy with the default. If you don't set this, the default is going to be the generalized, you use the generalized eigenvalue problem, because you solve the generalized eigenvalue problem, and that is usually an acceptable way to do the normalization. Again, have a look at the paper and preprint, and particularly supplementary material to discuss the different forms of normalization that you can use. And finally, the last one is the number of jobs. So allows you to use more than one thread on your computer. If you have a PC, if you have a laptop with multiple cores, say 4 cores, you can use all the 4 cores or 8 threads or however many you have there. Okay, so that's that. And let us try to see some data and let us try to run the analysis. Okay, so everything should be set up to be read by WBView, which is the Connectome project viewer. And we've done this because we think it's actually quite a nice viewer and gives very, very nice images. So that's the surface, right? We're just gonna have a look at here. So we have, that's the surface, and if we load a different thing here, that's the data that we're running, and give it some time to load. Here we go. And this is cortex left. Okay, so that's one of the surfaces, the very inflated surface, and we can see a time series, and we can go through this time series, and you can see every point. So this is going to be the input on which we're working on. Okay, this fMRI, resting state fMRI. Okay. So, there are some other files that I want to show you. We mentioned the cortical vertices. So the cortical vertices are just what we said they were, which if we just create, let's have a look. Okay, there you go. So the yellow is yes, part of the cortex, and the 0 values are no, they're not part of the cortex. And that's all we're interested in, is just a mask. And if we open the multimodal parcellation file, again, we have our parses, right? And again, the midline is 0. Okay, so that's done. We've had a look at the files. And so now let us run some analysis. I'm not going to wait for all of them to run because it will take forever. But I will show you how to run this. So VB tool. And we want our surface file, which is the very inflated surface. And let's see, we want the data file, which is the rest1 data. And we want output, let's just call it OHBM as an output base name. Right, and now we can do the optionals. So let's say we want to run the searchlight, and so for the searchlight we need the M file, which is the cortical mask, right, and cortical vertices, and that should be it. We don't really want to touch the norm, and we also don't want to touch the number of jobs because if absent, it will spawn one job per CPU. And I'm happy with doing that. And that should be it. The full brain is not needed because that is used for the full brain gradient, which we're not doing at the moment. We're doing the searchlight. So let's clear the screen first and restart. And you should get a little notification saying running searchlight analysis, and it will stop there until the next prompt comes, meaning everything is run. I'm not going to run this because my laptop is a little bit slow. So yeah, it's just terminated myself. So if you have a powerful, semi-powerful machine, it shouldn't take more than about a minute to run this. So that's using the doing the searchlight. So now let's see what we're going to do if we want to run the cluster. The clustered, we remove the M file, right? And we create the clustered file. So that is the multimodal parcellation. And if we do that, then that should automatically be recognized as a clustered version. And we can wait for it. Yeah, running ROI. We'll just cancel that. Running ROI analysis. So that was running searchlight analysis. And running ROI analysis, that will be the ROI. Sorry about this, it's just I terminated the process and you get all these errors. Okay, so that's how to run the clustered. And if you want to run full brain, you go to the same thing as if you're running a searchlight, but this time you put in the FB flag, and that should now run your full brain analysis. We can wait. Running full brain, this is the gradient analysis. And again, if I let this run, it will take forever. So, and it's only taking forever because this particular laptop is somewhat slow. Okay, so that's it. That's all you need to do to run the 3 types of analysis. And now let's look at some pre-baked results over there. Let's look at the file name conventions. So this bit is, well, whatever you put in. I mean, in my previous case, I called it OHBM, but when I ran this initially, it was called subject number, side, and whatever the resting states analysis was. I'd advise you to give some sensible names to this so that you can figure out what's happening. The next thing is the normalization. So GIG is the type of normalization that you use. And then you have 3 different versions. So you have this, sorry, let's start with this. This is the VBI, and VBI is the Vogt-Bailey index, so the searchlight analysis. The next one is the clustered, which is unsurprising. It's called cluster. And the third one, which is perhaps a little bit name, has a slightly strange name, is the whole brain gradient, which says whole brain, and then there's the normalization, and then it still says VB cluster. And the reason why it says VB cluster is because the whole brain analysis can be thought of as a special case of the cluster analysis, where you just have one big cluster. Okay, and now we will just run the WB view. So WB view, and we want to very inflated, and we want pre-baked, and we want to run everything in there. All right. Okay. So let's see. Okay. Okie dokie. So let us look at the VBI first, the VB index. And let us move that to percent so that we can see. And there we go. This is our VB index. You can see this is my preferred color map, but you don't have to use this color map. You can come here, and as you can see, this is thresholded at percent. The absolute thresholds, the highest value you can get is 1, and the lowest value you can get is 0. So you can see 1 to 0 over there. That's also quite useful. And we can roll through different color maps just in case you want to see how things look. Different color maps. So, right. So this is the VB index searchlight. Let's go back to spectral. I like that. Okay. The next is the VB cluster. And as you see over here, we have both vector and value. And the value gives us the VB index. So there we go, the VB index in each cluster. Right? And again, for an interpretation of what the VB index tells you, I will refer you to the paper so as not to make this video any longer than it needs to be. So that's the VB index. And we can have the gradient map now by going to the vector. And as you can see over here, you'll have the different parcels. I will show you what this looks like. So if you just move through each parcel, you will just find different parcels, and you will see the relationships within that parcel. So over here you have a gradient that goes from here to here. And you can pull out the VB index to see how how distinct are these two sort of intra-cluster regions, if it were. Okay, so that's the clustered. And finally, we have the whole brain. Open file. Ooh, we actually already have the file. Let me just show you this as well. The whole brain also has two. There's vector and value. The value just gives you one value, which is the VB index for the entire brain. So it's not terribly useful, but it's some sort of file nonetheless. And the whole brain vector is what we're after. This gives us the typical gradient maps. And that's it. That's all you need to do to run VB index. Thanks for watching. If you have any questions, just find me at the OHBM. You can find me on Twitter. My handle is my name and surname, Claude Bayada. And yeah, I'm looking forward to the conference.